Hello student, today we talk about the immune techniques and the topic is antigen antibody reaction from your chapter number 8 immunology. So basically we know that what is meant by the antigen and what is meant by the antibody. So let's see some their properties and their interaction response against each other. So basically, antigen is a kind of toxic material which is secreted by the pathogens such as viruses, bacteria, or foreign material. This antigen is kind of harmful for the organism's body because they can be produce the di- sorry they can be produce the diseases in the organism's body. Same along with this, for the elimination of this antigen, body have the defense mechanism which is called as immunity. This immunity having two types, that is innate immunity and adaptive immunity. Innate immunity is the first line of defense in which they act like a barrier for the pathogen for the initiation of disease causing. But if this innate immunity fails to eliminate this pathogen, then it triggers the phagocytic process. Phagocytic process in which cells phagocyte or engulf this pathogen and try to eliminate them from the organism's body. If the number of this pathogen is more than the limit of the phagocytic cells, then these phagocytic cells represent this antigen towards the adaptive immune system. Adaptive immune system is a third line of defense, because second line is a phagocytic cells. Third line of defense, that is adaptive immune system. In adaptive immune system, two kinds of components are present, that is T cells and B cells. T cells, which are the cytotoxic cell, that is they kill the pathogen and eliminate them from the body, where B cells are produce the antibodies. These antibodies are able to eliminate the antigen and pathogen from the organism's body because they fight against this antigen. These antibodies are mostly immunoglobulin protein of Y shape, which containing the light chain and heavy chain, and also which containing the variable region and constant region, where the variable region can be vary from their structure to structure. That is because human body containing different types of antibody, and this lots of diversity of these antibody are essential for the elimination of antigen, because in our environment there are our surrounding there are lots of kinds of antigen or disease causing organisms are present, such as helminths, bacteria, viruses, insects. So these kinds of organism, these kinds of microorganism harmful for the organism's body, that is human body, and we have to protect our body and make it healthy by using the immunity, by building the immunity, and these antibody help us to eliminate the antigen. Antibody is a kind of immunoglobulin protein which bind with the antigen, form antigen and antibody complex, which is also known as immune complex, and eliminate them from the body. So this antigen and antibody reaction mostly occur in the organism's body. It is similar to the enzyme substrate reaction. So their locking key mechanism resemble to the lock and key mechanism. We know that the what is meant by the enzyme and what is meant by the substrate. In biochemistry we study the enzyme and substrate. So during formation of product, enzyme and substrate bind to each other by the lock and key mechanism. The structure is quite similar for their binding. So one act like a lock and second act like a key, and they make a complex and form the product. Same as it is antigen antibody having the complex to form the lock and key mechanism. Antigen having the epitope and antibody having the paratope. These antigen and antibody form the complex which is called as the immune complex. This immune complex helpful for the removal of antigen. This reaction or formation of the immune response is irreversible reaction which is cannot reverse for the any kind. So this is irreversible reaction and having the non-covalent type of interaction. So for the binding of this antigen and antibody to each other, they having the non-covalent type of interaction. So there are many bonds are interact for the formation of the this antigen and antibody complex, that is immune complex. So for the detection of presence of either antigen and antibody, we use the lots of immune techniques for the detection of these kinds of antigen antibody in the organism's body. So identifying the molecules of biological and medical sensitivity by using the antigen antibody reaction. So basically antibody is a humoral response which is given by the adaptive immune system. For the antibody production, what happen? Basically, if innate immunity fails to eliminate the antigen, then it trigger the phagocytic process. Phagocytic process occur when it is limited number of the pathogen. If the number is beyond the capacity of phagocytic cell, then the phagocytic cells like a macrophage represent this antigen towards the T helper cell or adaptive immunity. So this T helper cell gives two response. First of all, he try to trigger the formation of T cytotoxic cell, and secondly it trigger the B cells. B cells form the two kinds of cell, produce the two kinds of cell, that is B plasma cells and secondly B memory cells. Plasma cells able to form the antibody and memory cells to produce the memory of this antigen. They having the memory of this antigen and antibody having the response against this antigen and eliminate them from the organism's body. So how this immune complex is formed? So antigen antibody, whenever they brought together and bind to form the complex which is called as immune complex. So whenever innate immunity fails to form to eliminate the antigen from the organism's body, then adaptive immune system trigger for the formation of immune complex and formation of the antibody. These antibody are specific for the antigen because the epitope of antigen is bind to the paratope of antibody and they form the immune complex. So in this process you can see that this is the cell. So this is the cell membrane in which you can see that this is the cell which showing the phagocytic process. During phagocytosis, they degranulate the antigen and this fragmented antigen represent towards the adaptive immune system by the macrophage cell, which is also known as the antigen presenting cell, APC cells. So this antigen presented towards the T helper cell. T helper cell then trigger the formation of B cell, that is plasma cells, and B cells differentiate into the two kinds of cell, plasma cells and memory cell. Plasma cell able to form the antibody. So in this figure you can see that the antigen bind to the antibody. So this antibody bind to the epitope of antigen and antigen bind to the paratope of antibody and they form the antigen antibody complex, which is also called as the immune complex. So the specificity of antigen antibody reaction occur due to their structure. So basically antigen can have the different kinds of epitope. Same as it is, antibody can also produce the different kinds of paratope. For example, immunoglobulin M. Immunoglobulin is pentameric antibody which can simultaneously bind to the 5 types of antigen. So this specificity of antigen and antibody reaction occur due to their structure because we know that the antibody having the different kinds of structure. First of all, they having the Fab region and Fc region, where the Fab region means the fragment of antibody where the antigen is bind. This Fab region having the two kinds of chain, that is light chain and variable chain. light chain and heavy chain. So this light chain and heavy chain also containing the two kinds of region. First of all, that is variable region. This is variable region and the constant region. Constant region, that is Fc region. Fc region is a fragment of crystallization which bind to the receptor of the cells, and this Fab region bind to the epitope of an antigen. So the binding site of antigen and antibody is the epitope of an antigen bind to the paratope of antibody. So antigen containing epitope, which is the binding site of antigen, and paratope is a binding site for antibody. So whenever these antigen get invented in our body system, antibody trigger for the reaction and formation of the immune complex. So this paratope is having the lock and key mechanism for the binding to the antigen. So similar structure. So this lock and key mechanism for the production of the immune complex is responsible for the production of the immune complex. So there are many types of forces are required for the binding of antigen and antibody. So there are many factors are also responsible for the binding of antigen and antibody. First of all, closeness between the antigen and antibody. So basically antigens circulate in the body of organism through the blood, and the epiphysis cells represent these circulating antigen towards the lymph node. Lymph node containing the B cells where they produce the antibody. These antibody and antigen are form the immune complex in the lymph node because they having the closely between their distance between on the cells is quite close. So they form the immune complex, that is antigen antibody complex. Also their intermolecular forces, because they having this structure which is capable to produce the immune complex and having the lock and key mechanism. So antibody and antigen having the structure for similar towards each other. So they producing the intermolecular force which attract towards each other and form the immune complex. So the also the factor, that is affinity of antibody. Affinity of antibody is higher, then it produces the high affinity towards the antigen for the binding. So this antigen bind to the high affinity of antibody and producing the immune complex. So also there are many forces are responsible for the binding of these antigen and antibodies, such as electrostatic forces, hydrogen bonding, hydrophobic bonding, and van der Waals bonding. So these forces are required for the bonding of the antigen antibody to form the immune complex. So that is for today. So in this lecture we talk about the how antibody and antigen produce the immune complex, because this immune complex is responsible for the formation of this antigen antibody form the immune complex. This immune complex show the immune response against this pathogen which is secreted, such a kinds of antigen which is specific for the antibody. So our body able to produce the different kinds of antibody which is specific for the specific antigen and produce the higher affinity towards the such kinds of specific antigen and form the immune complex. After the formation of immune complex, they remove from the organism's body. So that is for today. So in this lecture we talking about the how antigen antibody complex are formed. So that is for today. Thank you.